What's up, you guys? Rex here. And that's another week of med school in the books. Things are continuing to ramp up and getting more and more exciting every day. Alright, so I want to first talk about that this past Wednesday, I'd say was sort of my first bad day of medical school. Now, I'm still living the dream. I can't have a bad day. But this was my first day where I sort of felt like maybe I wasn't doing enough or wasn't doing things right. And so it was sort of a long day of lectures, and I was sort of getting behind on lectures because I was prepping for anatomy lab. And so I went to anatomy lab in the afternoon, and I was like, I know I have this one anatomy drawing thing that evening. I also have a couple lectures still to watch, some notes to take. And then at anatomy lab, this was the first time my group sort of didn't get everything done we were supposed to. And by sort of, I mean we very much didn't. That we had a very long dissection lab that day, and we're sort of split up into two groups. And my group of three was the second three. And so the first three maybe sort of didn't get as far as they should have, but then my group really didn't get as far as we should have. And so it sort of was my first time sort of feeling like I failed. And the real feeling of failure there was not oh, I didn't finish and I'm gonna, get in trouble, whatever. It's that oh, I missed out on this learning opportunity. Anatomy lab is one of my favorite things to do, and I want to get to dissect and learn by feeling and touching as much as I can. And so I really felt like I was failing myself and sort of failing my group mates a little bit and failing my other shift that they expected us to finish so that they could see what we dissected. And so we really didn't get there. And then it was sort of, walking back from lab, I was letting myself feel bad about myself for a little bit and sort of pity myself if I, should have done this, should have done this. And, maybe anatomy lab's harder and I'm not as good as it as I thought I was and all that kind of stuff. And then there's always that little straw on the camel's back of where I had to run back to anatomy lab through the hospital to pick up this one thing and walk back again. So it just sort of gave me more time walking to think and feel bad for myself. But the key thing that helped me get past this is I didn't let myself dwell on it. You gotta let yourself acknowledge it. I think that's important to say yes, I am disappointed in myself. That's why I sort of am feeling crappy right now, or I'm disappointed in this life event or that life event. In my case, I was letting myself be honest that I was disappointed in myself. I didn't do as good as I'd hoped I would, or as I think I should have, or any of that stuff. But I let that stop there, and that it became, all right, Thursday's coming. We're gonna hit it hard. I'm gonna finish these lectures. I'm gonna feel good about the progress I've made. And then Friday's coming. That was my next anatomy lab that I am gonna be overly prepared for that. I'm gonna be ready. Scalpel gonna go vroom. We're cutting things. We're learning things. We're feeling things, and I'm gonna kill it. And so I was really determined that Wednesday night I was gonna finish all my lectures and get ready for Thursday, kill Thursday, and Friday was gonna come. I'm gonna dedicate extra time preparing for anatomy lab, gonna kill it. And so thankfully, that's what I was able to do this week. But I know there's gonna be weeks that happen where I maybe have a bad day on Wednesday and I think I'm gonna make up for it on Thursday, and maybe I do worse then. And so I'm hoping that I'll be resilient. I've been thankful that I was resilient this first roadblock, and I think a big part is you practice on the little roadblocks, that you learn and be determined to know how to be resilient when you have those small hiccups, and that prepares you for the bigger ones. And I know the bigger ones are going to come as med school gets harder and I make bigger mistakes and I fail other things, that it's going to happen. But for right now, I'm thankful for it. I'm thankful that I had my first bad day. I learned from it, and it motivated me for the rest of the week, and I'm excited for next week. But so other than the one bad day, another great week of living the dream. It's definitely getting busier. We're having more and more lectures. I think we had 6 lectures the one day, in addition to a little 2-hour interactive group thing. And so that was a long day, but it's still at the point where I'm loving it. It's a long day, but I'm doing a long day of what I love, and there's not really something else I'd rather be doing. And so it's hard, but I like hard, and so that makes it easy. And that's a big key to making what you're doing easy, is if you really like it and you like that it's hard, you admit that it's hard and say that's a great thing about it, and therefore it'll make it pretty easy. But so enough about the week. Something I learned this week. So if you're new here, going forward, every Sunday, I'm going to share something that I learned in the previous week of medical school, hopefully in sort of a relatable way that you could say, wow, that's cool. I'm excited to learn things like that too. So this week, I got to see sort of 2 numbers you don't see every day. I got in 2 different contexts. I got to see some exponents that were 10 to the 150. Now, To give some perspective, there's like 10 to the 77 atoms in the universe. 10 to the 150 is not a number you see every day, or pretty much ever. I don't remember the last time I got to see 10 to the 150 in a lecture. I actually got to see some huge exponents in two different contexts. The first one is protein folding. And so there's something really cool that proteins are able to do is that they start off as this huge chain of amino acids. And you just think of them as a string of like letters that are all linked up, and each individual letter can be slightly different folded, and this aggregation of all these different little folds of all these different letters ultimately takes this complicated string of little amino acids and forms some even more complicated blob that does its function as a protein. And there's this really famous experiment, which is pretty much actually like a thought experiment, of that someone sort of calculated that if a protein were to try randomly all possible conformations, it would have to go through like 10 to the 150 different possibilities for just a medium-sized protein. And so even if it's trying like 10 to the 8 different possible arrangements, usually called conformations a second, it would take until like the sun consumed the earth for a single protein to ever find the right shape. And so from this experiment, we've come to learn that it's sort of usually visualized as a funnel, that one shape brings it to a closer to its low energy state where it's totally folded, and that moves it down. And so it's sort of like a funnel of different shapes that come down and point to the correct shape. But also very important to this process is something called chaperones. And so chaperones are other proteins that are actually super common inside the cell, and their job is to help and encourage proteins to fold to the right shape. And so there's different times where a protein without chaperones might literally take 2 million years to get to the right shape. But then you add in these proteins that encourage it to get to the right shape, and it'll take a quarter of a second. It happens almost instantly or faster, and it just works really, really well. And so sort of we then talked about, and someone raised this question, which I thought was a very interesting question, is that these chaperones are also very complicated proteins, and they need chaperones to fold them. And so it's an interesting chicken and the egg problem that I'd love to know exactly how this evolved and came to be and what mechanisms, that these chaperones, in order for them to work, need chaperones to fold them. And so these other chaperones that are folding the chaperones, they don't really have any purpose other than folding chaperones. And so did this original chaperone evolve first to fold the protein, which that protein, what the heck did it do without a chaperone to fold it? And then this chaperone folding a chaperone, what did it do before it had a chaperone to fold? So why would it evolve? And it's sort of a little circular logic of chaperones need chaperones and proteins need chaperones, but chaperones are proteins, and so therefore they need chaperones. So which came first? How did it exactly iterate through steps of starting with a very simple protein, and then it got a little more complicated, and it still did something to provide an evolutionary reproductive advantage to some cell, and then it got a little more complicated, then it started folding in a certain way, and then a different thing evolved and helped it fold. And so there's iterative steps, but it's still something that I would love to learn more about. And so there's another thing that's very similar, and that's enzymes and how they catalyze reactions. So enzymes are essentially a protein that does something similar of protein folding, but rather it helps a reaction take place. And it does this by selectively binding and sort of encouraging a little transition state of you have some chemical A and it has to turn into chemical B, and in between there it has to go into this high energy state where it's sort of in between A and B and then go down to B, and then the reaction happens. An enzyme sort of takes this little in-between state and it binds it in a special way that makes it much more stable and low energy, and so there's less of a hill. It doesn't have to go up from this energy here at A and get really high in energy in AB and then come back down to the B state, and now you have molecule B. An enzyme sort of smooths it out and binds this AB state. And the effectiveness of this is incredible, that some enzymes sort of speed reactions up like 10 to the 23rd fold. And so that is the difference between like a tenth of a second and millions of years. And without this, there's some reactions that would literally take longer than since the Earth has existed to ever happen. And so that's again sort of a chicken and the egg thing of how this evolved, is that this reaction, if it does happen, obviously confers some reproductive advantage to a cell that makes it more likely to pass that on to its other cells and they're going to reproduce more. But this will never take place without the enzyme. And so the enzyme needs to be there first. And so if the enzyme is there first, and the enzyme's real only purpose is to very selectively bind this specific reaction, what the heck was the enzyme doing there in the first place without the reaction to take place? And so it definitely happens in very iterative steps of that one reaction is able to take place but only a little bit. And so the reaction is there first, and then an enzyme evolves, and it makes it go a lot faster. And it definitely is possible that that's how it happens in very slow iterative steps, and we've got billions of years. But it's interesting to me that we like to think of cells as happening very fast, but there's also something happening inside of a cell. If things don't go right, there's something so slow that could happen without this enzyme that it would take on the order of billions of years. And so this reaction obviously needed an enzyme to first happen. And so I'd be curious to learn about like a specific example of what a reaction pathway looked like originally, sort of before the enzyme evolved, and then how the enzyme changed that reaction, and then how that reaction happening changed the enzyme to make it more efficient, and just a very complicated loop that happened over millions or billions of years. And so I find that very interesting that you don't get to see those huge numbers that are just impossible for your brain to comprehend. I can't comprehend something on the scale of the number of atoms in the universe or on the order of 4.6 billion years. And so it was cool to learn about. It, got me interested, got me excited, and that's what med school's been all about for me. Getting me interested, getting me excited. And if you want to hear more about that and hear more about my journey, make sure you subscribe, hit the notification bell if you want to join me along my journey. If you like the video, like the video. Dislike the video if you dislike the video. Any questions, comments, or concerns, I read and answer every one. I'd love to hear about them down below. That being said, don't be ordinary. Go be great.